You are listening to the Effective Statistician Podcast, the weekly podcast with Alexander Schacht, Benjamin Pieske, and Sam Gardner, designed to help you reach your potential, lead great science, and serve patients without becoming overwhelmed by work. Today, we are talking again with Sam, and we are talking about the second part of non-clinical statistics. Today, we are really focusing on what that means for research and development. So stay tuned for another really insightful episode. If you haven't listened to the episode last week, I would say first head over there and listen to that one first, because this is the second part of it. You can find out lots of help about visualization, leadership, better presentation, and all kind of other things on the homepage. We are not just publishing the podcast episodes there, we are publishing lots of additional things there as well. And also on LinkedIn, follow me on LinkedIn and you'll get much more resources. I'm producing this podcast in association with PSI, a community dedicated to leading and promoting the use of statistics within the healthcare industry for the benefit of patients. And the flagship event of PSI is coming up, the Psi Conference. Head over to psiweb.org and learn more about this conference. It's awesome. I am actually presenting as well. And there's lots of other great presenters. I think, just from a quality of the presentations, this is one of an outstanding conference. They always have much more people that want to present than they have slots. So they can have a good filter for high quality presentations. Check that out. I'm pretty sure you'll love it. And this time, it's virtual. You don't need to travel and spend thousands of dollars for traveling and hotel and all these kind of different things. It's very affordable. Awesome opportunity. And there will be lots of social interaction as well. Yes, there's a nice tool that PSI is using called wonder.me, and that enables us to have really nice social interactivity as well. Welcome to another episode with Sam. You probably heard the last episode where we already teased a little bit about the one today, where we will talk also about quality by design, which is a really cool term because it basically means that, you can improve the quality by working well with your statisticians. So you have all the right development of everything in a good shape. So Sam, how are you doing today?
I'm doing great, Alexander. I hope you're doing well too.
Yes. It's always fun to speak about these things, where, to be honest, I don't have so much knowledge about. So I'm learning quite a lot by just doing these podcast interviews, which is a lot of fun. So let's talk about R&D and about the non-clinical sides of R&D. Where does it all start?
Well, it starts all the way back into the area that you typically call discovery. So discovery is where you're trying to actually find a new molecule or antibody that has some therapeutic benefit. And so there's a lot of work that happens on either engineering these new protein molecules or finding a chemical structure for a molecule that would go after some sort of biological function. Mm-hmm. So an example might be if you wanted to control asthma, right? Well, there are certain things in your body that trigger an asthma reaction, and biologists and doctors, they learn those things. It's so usually some sort of immune-based activator, right? That activates the mast cells in your body and then makes it produce histamine and different things like that, right? Well, what if you can stop that somewhere along the way, right?
Yeah, it's this kind of long process, and says,
right.
Once stone hits over the next one, and
Right.
can you do some kind of barrier in between so
Right.
you know that the energy is absorbed of the stones? Yeah.
So you want to find a compound that maybe binds against the antibody that's in the blood that starts the chain, or maybe you want to find a small molecule that binds to the final end of the chain that prevents that final immune reaction to happen, right? There's different ways you can control and mediate those immune responses. And so you, in order to find those molecules, you honestly, you might have an idea what it is, but you have to try lots of different things and lots of different variations of molecules or variations of proteins. And you usually have to have sort of measurement method, like how well does it bind to a receptor site or something that looks like a receptor. And so you have to have an analytical measurement method, a chemical method, and you have to have a process that you use to do the testing. And it's usually a high throughput process. So you're doing hundreds of these or thousands of these, over a short period of time. And you're trying to screen out the ones that have a high signal. And if you think about that as a statistician, you know that, okay, that's a classification problem, right? You're making measurements. The measurements have error associated with them. And you need to make a decision based on that. And it's really a go or no go. This goes to the next stage of development or not type question. And so, and you've got different types of errors you can make there. You've got the type 1 error, which is you move forward a molecule that you shouldn't, right? Because it looked like it was a good signal, looked like it could have good binding activity, but it really didn't. And the second one is the type 2 error is you miss something that you should have moved forward. And so where do statisticians help in that area? Well, they help with, one, making sure those processes that do the testing work run well, right? The test methods are appropriate, that they have the appropriate level of variability, and that the risk associated with using those methods, the variability creates risk. Those type 1, type 2 risks, that those are understood and managed in an appropriate way. So it may be what you do is to avoid the type 1 error, you come up with a sort of a statistically based decision rule where you say, okay, it passed the first level screening. Now I'm going to do a second level screening with replication. And with more replication to confirm that second result. And if it goes through that stage, then it may be a better candidate. Kind of like the phase 1, phase 2 clinical trial type approach. But instead of just, one trial, you've got hundreds and thousands of these coming through, right? Another area is they have these libraries of information about molecules, and you can quantify a molecule structure based on, the angles of the bonds and the length of the bonds and the molecules that are in there. And you could think about it, you could code that like a big vector, right? That's just of the molecular structure. Well, if you have this big vector, and it can be a really big vector, think about it for a protein, right? It's huge vector, thousands and maybe millions of elements in that vector. Well, you want to do some data mining on that. You want to say, I know in the past molecules like this had good activity and these other molecules didn't have good activity, but now I've got 20 times more molecules that I just made up in the computer that I simulate, that I haven't actually physically made yet. Which one of those might be a good candidate based on their potential structure? And you go through that process to say, oh, these 100 new molecules might be good candidates. Let's go make them. So it's more of a predictive modeling type problem. People who do bioinformatics get involved with that sometimes too. So it's those are some interesting problems. Some are just more on just sort of traditional, what I think we call quality in the process that you're using to make decisions. And the other one is the data mining aspect of it. And there's probably more to it as well. I haven't worked as much in that area. Maybe we could find somebody who knows a little bit more about that sometime and have them on the podcast to talk about what it is they do in the area of discovery.
Yeah, but I can see that there's potentially you have the problem that your vector of all the variables is much bigger than the sample you actually have. So, and that poses lots of interesting problems, Usually in statistics, it's the other way around. Yeah, you have a couple of endpoints and then you have lots of subjects or samples from it. and
Yeah.
not the other way around.
High dimensionality is something that we as statisticians should know how to address appropriately. And that's one place we really provide value, when we have those really wide tables with not many very rows in them.
Yeah, yeah. So that is dimension reduction approaches. Anything that you have particularly used for dimensional reduction?
Well, in the situations where I have done that, and this would be more on the manufacturing side of things, where you have lots of information about the manufacturing process, and maybe every batch of material you make, you may have 100 or 200 columns of information, for that one single batch. And I've used principal component analysis and partial least squares, regression. Some new ideas that are out there that are useful are things like regularized regression using the lasso or the elastic net. Those are also pretty valuable, at least for maybe identifying what are those really important factors that might be driving the output. And then if you want to make predictions too, you can do that as well. I've even used stepwise regression sometimes.
Okay. So for the principal component analysis, it would be basically if you think about the covariance structure, you try to simplify that and looking into, combinations of linear variables that drive most of the variation. And that way you can, look into only a couple of combinations of these variables rather than to look into each of them. But the regression analysis would be more like you have the dependent variable, and then you have lots of independent variables, and then you try to reduce the number of features in your model, so to say. Yeah. Right.
Or you combine both of those. You can actually combine the regression with those dimension reduction ideas that principal components uses. That's what partial least squares actually does.
Okay.
So principal components, what it does mathematically, if you have the x variables in your model, y is equal to a function of x, you find the directions in that vector, in that space of those x variables that has the most variation.
Yep.
And that's your first principal component.
Yep.
And then for principal component regression, you would say, okay, let's take that first principal component. Let's do this transform and convert, 100 columns into one column by taking a linear combination of those 100 columns. That's your transformation to get your first principal component. Then you say, is that correlated with the response? And you fit a model. Well, partial least square says, okay, let's find the direction in that X space, the design space of your problem. But let's do it in such a way that it captures a lot of variability in that X space, but it also at the same time is highly correlated with the response.
Yeah.
So it's more of it's a different type of optimization problem to get to that answer. And, but it ultimately, it just uses things like eigenvalue, eigenvector decompositions of the data, and then you get the right answer out. So, and software does it automatically too, which is nice.
Yeah. Welcome back to linear algebra 1 and 2.
Right. Yeah. Sorry. If you didn't like linear algebra, or you're just listening to hear what podcasts think, or what statisticians think about on this podcast, sorry for all of that math. So.
Yeah. Yep. And if you're coming from a completely different background, then just try to understand what we just said in terms of kind of, verbalizing this, is that you either look into the covariance structure, or you look into also the variable covariance structure with the endpoint you're interested in. So the Y. You can combine both of that. So that's then the last method that we talked about. Okay, very good. Let's go into the next phase. After discovery, we get into early phase, and there is the first species coming into play.
Right. So one area where you get involved with the non-clinical statistics, and I think for human pharma, non-clinical means not clinical trials in humans.
Yeah.
Right. But you do trials and studies in animals. And that's often handled by people that are more in the non-clinical area, particularly things that are related to toxicology and pharmacology. So toxicology is, will this molecule harm me? If I get it into the body system, will it cause harm into the systems in my body? Right? That's what toxicology is. It toxic. Pharmacology is, well, what happens to the molecule when it gets in the body? Where does it get absorbed? How fast does it get absorbed? What parts of the body does it get distributed to? Is it metabolized in the liver first? Those types of things. And so statisticians really collaborate a lot with toxicologists and pharmacologists on understanding the systems. There's actually a whole statistics area around compartmental modeling, which really you model the body systems as compartments where you have flows in between the compartments, and there's a differential equation that describes that flow between the compartments. And then if you understand that, then you can end up fitting models based on data that you collect to estimate those rates of moving between the different compartments.
Yeah, it's a little bit like if you think of a big organization and you have, put some pressure on it, and then, this pressure gets through the different departments of the organization and they interact with each other, and, finance might do this and HR might do that, and that might cause sales to do something different, and that has a back effect on finance again, and so on. So the same basically happens in your body.
So they use animals to do that, and they use an animal model often to measure those types of things. And where do statisticians get involved with that? Well, one is just the study design. What's the appropriate number of animals? What's the randomization that you should use if you're testing against a control? And oftentimes you have interesting randomizations. It's the randomization might just not be pure randomization. There might be some blocking effects that have to happen where maybe it's the pen or the room that the animals are stored in. You want to block on those because there can be environmental effects on the animals as well, and you want to control for those. So statisticians get involved with the study design quite a bit, and then subsequently the analysis of the study after it's done.
So in terms of blocks, that's actually an interesting part. I know it from clinical studies that, your blocks are within, for example, a physician. Yeah, so you have different physicians and you would randomize within these different physicians. Yeah, and let's say you have 2 treatments and you have a one-to-one randomization. So for every patient that gets treatment A, you have 1 patient that gets treatment B. You could have, let's say, a block size of 4. Yeah, and so within this block size, there's 2 with treatment A and 2 with treatment B. And so basically you can have, AA, BB, or AB, AB, or BABA, or BAAB, or BBAA, I guess.
You're very good with those permutations there.
And so this is kind of when you want to make sure that within these different blocks, you have always more or less the same number of patients on both treatments, because these blocks can have an effect. And if you then have certain blocks that only have A and other blocks that only have B, then you cannot understand what the block effect is versus the treatment effect is. Yeah, so because both are confounded with each other. And I guess here it's the same. If you have, for example, 2 different labs or 2 different, types of animals in whatever type, one is coming from one parent animal and the next one is coming from another parent animal, or different days or whatever.
Yeah, and a lot of it is just understanding what are those nuisance sources of variation that you don't want to include as variation to attribute to the effect that you're trying to measure. So you're just trying to avoid nuisance variation from having you make a mistake in the ultimate decision that you want to make.
Yeah. So in terms of this blocks, of course you can have, lots of blocks, but there's then a limit to that as well. Yeah, because then you end up having just, 1 or 2 subjects. And this
Right.
has animals per block, which is also not helpful.
Yeah. And so you have to try to find the right balance between the information you gain from the experiment and the amount of resources you have to invest to get that information. And for a lot of reasons. Sometimes it's just time and money issues. There's also animal welfare issues. If you're going to do experimentation on animals, you don't want to do it more than you need to, right? Because we want to use the animals for the purpose they were, bred for, but we don't want to waste them, and we don't want to unnecessarily sacrifice those animals, because most of the time the animals are euthanized after they've been put into a test. So there is that aspect of it. So yeah, there's a lot of issues there where you're trying to balance you'd be great if you said, well, I need you to test 100 animals. And you said, well, don't you understand these animals cost $200 a piece and it's going to take me 3 months to test 100 animals. Yeah. Well, I can't do that. So you have to find the right compromise there to say, well, what's the number you need to test to get an answer that helps you make the right decision?
Yeah. And that's an interesting problem in itself. Yeah. Mm-hmm. To understand kind of how you design the experiment has the design of the experiment, the experiment itself, has some kind of operating characteristics. Yeah. So, and understanding that is important. For that, you need to have clear what are the goals that you actually want to achieve. Yeah. And not otherwise think about, now we have done the experiment. Are we now more knowledgeable than before?
And I'll have to admit, I don't know that area as well as I do some other areas in R&D. I just know about it because I've been in R&D a lot, and I have heard about it and seen what other people do. Again, it'd probably be good to get somebody that knows about this area a little bit better, who's a statistician working in that area, could talk about it in a little bit more detail or with a little bit more clarity than I provided. So.
Yeah. And that is a good topic for another podcast.
Right.
Okay, what else happens in early phase?
Well, now I'm going to switch over to what I know really well, which is sort of that mid to late phase development, and where you're trying to develop the chemical formulation and the chemical process to make the drug product. So you really have to start early on by what we call process definition. You define the process, and you work on a team with chemists and engineers and biologists and a broad variety of people, even regulatory scientists, people who understand the regulatory requirements really well. And you come up with, okay, this is how I think I should make the medicine. Let's try it. Let's do some trials to see if we can make it relatively consistently at this target, and then make material that can be used in clinical trials with that target production process. Mm-hmm. Okay, so that'd be a big important milestone that has to happen in pharmaceutical development. And if you can get involved early on in that as a statistician and influence that by having people do more structured experimental design around that process definition step, that process definition overall process, right? It's a process to develop a process. So, So the process to come up with the target for production that you use, that overall laboratory experimental process, if you use good principles of statistical experimental design along the way, you're going to advance the development of that drug product process much more quickly. And so that's where I've had some good success, where normally people might just try like these one-of experiments. Let's try this today. Let's set the temperature at 100, and let's set the pressure at 20, and let's set the reaction time at 2 hours, and let's see what happens. And then the next day they say, it didn't turn out so well. Let's increase the temperature. Let's see what happens, right? And they just sort of fiddle around and just try different things. And what I'd like to do is say, well, let's think about it. What do you think might influence this chemical reaction? Can you list out the 3 or 4 or 5 things that might be influencing it? Usually the chemist or the engineer can sort of say, yeah, I can list that out. And then, well, how much experimentation do you want to do? He said, well, I'd like to get the answer in a week, and I can do like 4 reactions a day. So I can do 20 reactions. So let's see, I've got 5 factors. I can do 20 experiments, and they're 4 per day, right? Well, that you can then use principles of experimental design to find a good experimental design that fits within that experimental plan, right? The experimental resource plan. Yeah. And you typically use some sort of software to develop the design, right? That does it for you, where you give it the parameters of that I described, and it'll say, this is a design that will allow you to understand what's important, what affects the output of this chemical process, right? This chemical reaction. Is it temperature? Is it pressure? Is it reaction time? Is it the ratio of the solvent? Those types of things. And then you can get a target by having an equation that makes a prediction, right? You do an experimental design, you generate data, you fit a model that makes a simple prediction about what the quality is going to be of that material. And then you use that equation to find an optimum or find a good target. That's really kind of experimental design in a nutshell in that area, right? That's how you apply experimental design.
So basically you have something like, okay, the chemist says to you, okay, we'll test kind of temperature between, let's say, 20 and 40 degrees. We'll look into pressure in that range. We'll look into these different dimensions for the ingredients. And then you come up with a design and say, well, on the first experiment, you set the temperature here, the pressure there. So you use these kind of ingredients. And then in the second one, you do this. And in the third one, you do that. And when you have done all your 20 experiments, you come back to me, and then we model what the overall kind of experiment has told us. And then we can say, oh, you can forget about the temperatures. That's, within that range, nothing really happens. But with pressure, there you need to take care that, within this kind of bar range, you need to have a little bit more closer look, because there's something is breaking there. Yeah.
Right. And so the idea around it using a statistical experimental design is when you've got those multiple factors that might be influencing this chemical reaction, you want to do the variation in those factors in a way that you don't confound their effects. Yeah. So you don't, I think I described that earlier on, right? You don't move temperature and pressure always in the same direction, right, at the same time, because then you won't be able to separate out if something changed in the output. Was it temperature or was it pressure? So, and as you get more factors, you need a better experimental design to account for that. Yeah. What really makes this tricky or complicated in this area, what I just described for a typical new drug product, those types of problems happen dozens of times. For different parts of the process. Because each of these processes have multiple steps involved with them. Each step needs that kind of focus, right? To understand how can you make a target for this process. And then you have also all of the quality methods that are used to test the quality. They're being developed at the same time. So you're making this product while at the same time you're making the ruler that's going to measure the product. Yeah. Yeah. And it's and those have to sync up together, and you have to make sure that those analytical quality methods that are being used to measure the quality of this material are pretty good too. And that's just to get ready for clinical trial manufacture. Yeah. Okay? Yeah.
You basically have then a design space where you have actually lots of missing cells, so to say. Yeah. You have, actually, by far, most of your cells don't have any observations in it. Yeah, so you have only a couple of observations in this big space, and then you try to do some modeling in it.
Yeah, it may turn out that the best way to make a drug product is the way that you never tried, but you did enough experimentation around that to know that, oh, here in this area where I didn't do an experiment, that should produce material that has the desired quality characteristics that I want it to have.
Yeah. And so you can do a follow-up designs and
so after it goes to clinical trials, then the whole next phase of development starts is, okay, we're hoping this clinical trial is going to be successful, right? Because statisticians like you and that work in the clinical area, you by this time you're assuming this is probably going to be successful, and we're going to spend all this money to do a phase 3 clinical trial, and we're going to design a really great study to do that and spend unreasonable amounts of money sometimes to do those.
Couple of hundreds of millions sometimes.
Yeah, lots of money. So we're kind of assuming that that's going to go forward. So now the clock is ticking. We've got to get the final process defined on how to make this drug product. And in the regulatory submission to get approval, when you submit this, you don't just submit a target. We got to make it with this temperature, this pressure. You have to submit it with ranges too. You have to say temperature between 10 degrees and 30 degrees and pressure between 1 bar and 2 bar and so on. And you've got to do that for every step of the process too. And there's just multiple decisions that have to be made where data is being collected to make a decision, and it's pretty complex. And so you really kind of have to understand not just the science, the physics and the chemistry and the engineering. You also have to understand the regulatory requirements. What are the regulatory agencies expecting to see? What type of data do they want to see, and how would they want it to be analyzed? The ultimate goal is to use this idea called quality by design, where what you've done is you've mapped out what are the key quality attributes that are important to the customer.
Yeah.
Right. The patient that's going to use this medicine. Map all those back to the properties of the drug product that impact those things. So, the patient wants the material, wants the drug when they take it to treat the disease. Right. So it's got to have the right potency. It's got to be safe. Right.
Yep. No side effects. Needs to be
no side effects.
Yep.
Clean, not broken tablets, things like that.
And so it needs to be stable. Yeah.
Stable. Right.
When it leaves the lab, or that until it reaches the desk of the patient, it's still the same.
Right.
Or probably still in the same range at least. Right. Yeah.
So you're using those principles of quality by design. You map those things back to things like, okay, well, I know when I make a tablet in the drug product, I can measure how easy it is to break that tablet. Mm-hmm. They have these little testers where they measure the hardness of a tablet. They try to break it and they see what force causes it to break. Okay, well then what's the criteria for the tablet hardness, the breaking force required to break that tablet, that ensures that when it gets to the patient in the bottle, it's not going to be a broken tablet? You have to figure that out. It takes a lot of experimentation to do that. That's just one aspect of the quality of the product you have to determine. You have to determine all of the specifications that are, so what are the attributes you're going to measure, and the criteria that those attributes have to meet to make sure the batch is of good quality. And then you have to also develop ranges and a control strategy that says, if I operate this process within these ranges, I'm going to make material that produces good enough quality. It has the quality attributes all meet these specifications that we develop, so that in the end, it's good for the patient, right? It's safe and efficacious for the patient. And there's a lot to it there.
So you basically need to, for all the steps in your development process, you need to have a good control of all the variables that are important. And you need to have, for all of these, thought through kind of experiments that would lead you, all the answers to all these different questions. So that if you, have all this kind of string of lots of different steps, that the overall quality of this very, very long string of stuff.
Yeah. Ideally, what you'd like is a model that makes predictions about the process. If I said, these are all the inputs to the process, I'm going to predict what comes out.
Yeah.
Right.
Yeah.
Unfortunately, the real models that describe these processes are pretty complex. So again, using statistical analysis, regression modeling along with good experimental designs, you can build good approximate models that let you make predictions. And the level of fidelity that those models needs, it depends upon how much risk is associated with whatever it is you're trying to model. If it's the final step of the drug product process, that's probably more likely to potentially cause a problem to the patient than maybe something that happened 10 steps back in the process. right? Because along the way, there are controls and purification steps and quality testing all along the way that would weed out the bad material that comes through early on. But that final step, that's got to be right? So you probably want a good model for that one if you're going to use a model to make predictions.
Yep.
And that's the dream. That's the idea around quality by design, is that you could actually have a model that predicts the batch you never have made yet. That's really what would be nice, right?
Cool.
And
cool. Okay, so quality by design. We did a lot of teasing in the last episode, and today you got a feeling of what that looks like and what it takes, and that it also includes a lot of knowledge of the space you're operating in, that it also includes lots of discussions with the other experts you're involved in, so that you understand what matters and that you understand when he speaks in his kind of scientific or functional area language, what he's actually talking about. So it's obviously also requires lots of communication skills. And so with that, thanks for listening today. And next time we'll speak about, what are the big differences between clinical statistics and non-clinical statistics, and what the different worlds can learn from each other. This show was created in association with PSI. Check out the homepage to register for the conference. Thanks to Rain, who helps with the show in the background, and thank you for listening. Reach your potential, lead great science, and serve patients. Just be an effective statistician.